Welcome to Planetterrian Daily, episode 29. Today is March 11th, 2026. European trial finds sulthiame drug cuts sleep apnea breathing pauses by up to 47%, improving oxygen levels. European trial finds sulthiame drug cuts sleep apnea breathing pauses by up to 47%, improving oxygen levels. Let's dive right into that lead story because if you've ever dealt with sleep apnea or know someone who has, this could be a real shift in how we approach it. According to Science Daily, a clinical trial out of Europe tested sulthiame, a drug that's already used for epilepsy, on people with moderate to severe obstructive sleep apnea. What they found is that it stabilized the brain signals that control the muscles around the airway, reducing those disruptive breathing pauses by up to 47% in some participants. That not only meant fewer interruptions during the night, but also better oxygen levels overall, which is crucial since poor oxygenation ties into everything from daytime fatigue to higher risks of heart disease and stroke. The beauty here is that it's a pill, non-invasive, no masks or machines like CPAP, which a lot of folks find cumbersome and end up abandoning. Researchers noted this could be a game changer for the millions affected worldwide, especially since the trial showed improvements in sleep quality without major side effects in the short term. Of course, it's not a cure-all. Longer-term studies are needed to confirm safety and how it stacks up against existing treatments, but it's fascinating to see a repurposed drug targeting the neurological side of apnea rather than just mechanical support. I've got to say, as someone who's seen friends struggle with this, the idea of better rest without the hassle is genuinely exciting. It's targeting the root brain signaling issue in a way that could open doors for more personalized therapies down the line. Shifting gears to something that's got big implications for food security in a warming world, let's talk about a breakthrough in plant engineering. Science Daily reports that researchers have pinpointed a protein from hornworts, a type of ancient moss-like plant, that helps cluster Rubisco enzymes more efficiently. Rubisco is key for photosynthesis, capturing CO2 to turn it into energy, but it's not always great at its job, especially in hot conditions where it wastes energy. By engineering this protein into other plants, they boosted CO2 capture and overall photosynthetic efficiency, which could lead to crops that yield more even as climates change and CO2 levels rise. It's an incremental step building on what we know about bioengineering, but it adds a clever twist by borrowing from nature's own toolkit rather than starting from scratch. Think about it for staple crops like rice or wheat. This might mean higher outputs without needing more land or water, which is vital as global populations grow. I'm intrigued by how this highlights evolution's hacks. Hornworts have been around for hundreds of millions of years, and now we're adapting their tricks for modern agriculture. Future research will test this in field conditions, but it's a solid reminder that small molecular tweaks can have outsized impacts on sustainability. On a related note in the ocean realm, there's new insight into how marine microbes are holding up against climate change. According to another piece from Science Daily, scientists studied Nitrosopumilus maritimus, a key microbe involved in nitrogen cycling in the seas. As oceans warm and iron becomes scarcer, iron being essential for their metabolic processes, these microbes have shown remarkable adaptability by optimizing how they use the limited iron available. This resilience means they can keep converting ammonia to nitrite and nitrate, which supports the entire marine food web from plankton to fish. It's not flashy, but it's critical because disruptions in nitrogen cycling could cascade into dead zones or collapsed fisheries. What stands out to me is how this refines our models of ocean ecosystems. We already knew warming stresses microbes, but seeing this specific adaptation mechanism gives us a better grasp on potential tipping points. For longevity folks, it's a nudge that planetary health ties into our own. Healthier oceans mean better air and food chains that indirectly support human well-being. Researchers suggest this could help predict how nutrient balances hold amid ongoing shifts, and it's worth watching how it plays into broader climate strategies. Now, let's touch on something a bit more cautionary from the world of cannabis research. Science Daily covered experiments showing that THC, the main psychoactive in marijuana, can induce false memories even at moderate doses. Participants who took THC were more likely to fabricate recollections when tested on memory tasks with disruptions across multiple brain systems involved in encoding and recall. This isn't just about forgetting where you put your keys. It highlights risks in decision-making, like in legal settings where eyewitness testimony might be influenced, or everyday choices affected by altered perceptions. Building on what we know about cannabis's effects on the hippocampus, this study adds evidence that it's not benign for memory integrity, especially with increasing legalization. I find it thoughtful because it doesn't demonize the drug but underscores the need for awareness, great for pain relief or relaxation, but maybe not ideal before a big meeting or court appearance. The findings could inform guidelines for safe use, particularly in therapeutic contexts, and it's a good prompt to consider how substances tweak our brain's reliability over time. Speaking of health interventions that could save lives, there's promising news for folks on dialysis facing heart risks. A trial detailed in Science Daily found that taking 4 grams of omega-3 from fish oil daily cut cardiovascular events by 43% in high-risk dialysis patients. These are people already vulnerable due to kidney issues where heart problems are a leading cause of death and options have been limited beyond standard care. The mechanism seems tied to omega-3's anti-inflammatory effects and how they stabilize blood vessels, reducing things like heart attacks or strokes. It's an extension of what we've seen in general populations, but tailored here to a group that really needs it, showing that a simple supplement can make a measurable difference without complex procedures. As a Canadian who's seen our healthcare system grapple with chronic conditions, this feels like a practical win, affordable, accessible, and evidence-based. Of course, it's one trial, so replication is key, but it opens the door for integrating this into routine dialysis protocols and perhaps preventing complications that shorten lives. Diving deeper into the cutting edge of gene therapy, 2 related studies in Nature caught my eye for their focus on improving delivery systems. One developed a lysosomal barcoding method to measure how well nucleic acids escape endosomes in the liver, essentially quantifying that tricky step where therapies get stuck inside cells instead of doing their job. By optimizing branched lipids, they boosted delivery efficiency for RNA-based treatments, which is huge for targeting liver diseases like genetic disorders or even some cancers. The other built on that with an in vivo assay that directly enhances nanoparticle escape in living systems, again refining hepatic delivery. Together, these aren't just tweaks. They provide tools to design better lipid nanoparticles, expanding options beyond what's currently used in things like messenger RNA vaccines. What fascinates me is the mechanism angle. Endosomal escape has been a bottleneck, and now we have ways to test and improve it in real time, which could accelerate therapies for everything from hemophilia to metabolic conditions. For longevity research, this means more precise gene edits that might extend healthy lifespans by fixing underlying issues earlier. It's incremental progress, but it sets the stage for safer, more effective interventions. Staying in the genome editing space, Nature also reported on engineered TNPB nucleuses that have been optimized through mutational scanning for use in plants and human cells. These are compact tools derived from bacteria, improved for precise cuts without off-target effects, making them versatile for agriculture like creating disease-resistant crops, or therapeutics such as editing genes linked to hereditary diseases. It's building on CRISPR's foundation, but offering alternatives that might work better in certain contexts, like hard-to-edit plant genomes. The population it affects could be vast, from farmers needing resilient varieties to patients with rare genetic conditions. I appreciate how this varies the angle toward future research. It's not a headline grabber yet, but it expands the toolkit, potentially leading to breakthroughs in food production and personalized medicine. Researchers note these could be combined with delivery advances we just discussed, so it's all connecting in exciting ways. On the diagnostic front, another Nature study found that elevated phosphorylated tau in the blood acts as a biomarker for systemic amyloidosis, specifically types like immunoglobulin light chain and transthyretin. This could help diagnose these protein buildup diseases earlier, distinguishing them from other neuropathies that mimic symptoms like nerve pain or heart issues. Amyloidosis often flies under the radar until advanced, so a blood test like this might enable interventions before irreversible damage, improving outcomes for affected folks. It's an addition to what we know about tau from Alzheimer's research, repurposed here for broader applications. Clinically, this matters for older adults where amyloidosis risks rise, tying into longevity by catching age-related declines sooner. My take is it's a thoughtful step forward. Reliable biomarkers are gold in medicine, and this one seems promising for refining diagnoses without invasive biopsies. Wrapping up with a look at cryopreservation, which always piques interest in longevity circles. Lifespan.io covered scientists who successfully vitrified and rewarmed mouse brain slices and even a full brain, preserving much of the structure upon thawing. Vitrification is like flash freezing without ice crystals that damage tissues, and they managed to keep neurons and connections intact enough to study post-thaw. This advances techniques that could one day apply to organ banking or even whole-body preservation for medical purposes like buying time for treatments. It's not ready for humans, of course, but it builds on prior work by showing viability in complex brain tissue, which is notoriously delicate. For those tracking cryonics or tissue engineering, this is an incremental win that refines the science. Fascinating to see how far we've come from basic freezing methods. Before we go, next time we'll be watching for updates on those optimized genome editing tools, as early trials in agriculture could reveal how they perform in real-world crop yields. That's Planetterrian Daily for today. If you enjoyed this episode, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow.